Thanks everyone for having me here. And I'll preface this by saying I should have remembered who I was going right after, but I think this is a good compliment. Mike, I'm thinking a lot about how we can take all the data that we have now and implement it in a clinical system that I fully believe and agree with it is broken. I want to think about precision medicine today and what our perspective is as researchers. But in order to think about that, I just want to recognize where we've come from, right? I remember sitting in the room with Eric Lander presenting when the Whole Genome Project came to completion, and the perspective that genetics alone was going to be the thing that really changed how we treated people, right? We would really find the accumulation of genes that would cause a lot of diseases and implement treatments. And, you know, when we look now, 20 some years later, we really see that while genetics is important and we have made advances, that really genetics alone is by far not the picture. And in fact, for most people, it's just a small percentage. As was just said, 16% is a genetic impact. But right, what we really have to appreciate is there are all of these things that happen: exposures, lifestyle, medication, everything, and how this impacts our health trajectory is really critically important to capture. And in thinking about this, I think there's another thing we can take from the Human Genome Project. And I read this quote and it has really stuck with me. And that's the success of the Genome Project as reflected by Richard Gibbs. Its success should be measured by how this project transformed the rules of research, the way of practicing biological discovery, and the ubiquitous digitalization of biological science. And I think that's really the perspective that we have now when we think about biological science and how we are going to improve individual life health journeys. And I always think about this in terms of where we really want to go, right? And when we think about this in the framework of electronic medical records, what we really want to do is completely change what we see and how we treat patients. And again, there are many factors of this, and I'm not going to be talking about all of the factors of changing from a diagnostic treatment-based reimbursement to a preventative reimbursement framework, and that is a huge part of it. I think changing it so we can have reimbursements by preventative medicine is a huge part of this. But in addition to that, what we really want to see, right, is we go to the health records and we look at a record, and instead of seeing what we see now, we have a lot of information that is collected on individuals based on all of these metrics of health, including the environmental exposures combined with the genetic backgrounds to give each person the individualized treatment that they receive. And as you could just see from the last talk, that is critically important, right? I really hope I'm a responder to cookies and that my blood insulin doesn't go up and that I'm not as responsive to banana. I am responsive to bananas. That would be great. But we want to know this, right? And so I want to talk about knowing this in 3 different frameworks and taking what Mike just presented about really dense repeated measures on a smaller number of individuals. I want to talk and think about how we can scale that up to a larger number of people in implementation in bigger settings, and what we've done there, and what we can understand, what we know. And you know, I'm going to talk about this with an example of using electronic medical record data as it is. Some of the things we see with omics, you know, there was a lot that was just presented. I'm going to be talking about this in a little bit of a different framework, but then going to multi-omics and thinking critically about how this actually gets implemented in the in clinical care. So in thinking about EMR, right, I am going to be talking about biological age as our primary phenotype, primarily because, you know, as was sort of just said, it's a very good metric of sort of cumulative health. And so, you know, thinking of biological age, we can think of it as this totality of health in many different perspectives, from heart disease to nutrition to things like grip strength and cognitive decline, et cetera. And recognizing that in this, there's both these combinations of nature versus nurture. And I will highlight that with this picture. So this is yours truly 23 years ago. I hope I look the same. And you see my great-grandfather here, who is 107. He lived in 3 different centuries, which is truly remarkable. But what is even more remarkable is that he was diagnosed with black lung in his 30s because he worked in the coal mines, and he chewed tobacco his entire life. So he's a perfect example of someone who did not do the right things, yet somehow, overcame that to live to be almost 108. And I truly hope I have his genetics, because that would be really great for me. But so when I'm talking about this with electronic medical records and biological aging, I'm going to be talking about it using some snippets from this paper that we just resubmitted to Nature Aging that has various metrics of biological age that I'm going to talk about. But one of the first things we asked is this idea of, okay, biological age we can see is super important, but is there a way we can practically implement that within the current EMR framework? And this is important because if we could do that, right, we can look at those aging trajectories, but not just look at it in a small number of people, but we could look at it in millions, right, and actually get these trajectories down really well. And so the objective was to create this clinically relevant and reproducible biological aging phenotype in an EMR setting. And so we called this metric EMR age. And what did we want it to reflect? Well, we wanted it to reflect 2 things, right? How long we still have, which is a super important thing, but not only how long we still have, but the quality of our life and our current age, right? Because we can have a long time, but if we have a really poor quality of life, that is not a good thing. So we did that looking in the Mass General Brigham Biobank, where we had 31,000 people that we followed up to 30 years. And again, looking at this metric of EMR age, we wanted to make this clinically reproducible measure of EMR age, balancing mortality with health and wellness. And so we purposely did this in a framework where we wanted the EMR age connected to our biobank that also had biosamples, because when we think about the omics, right, we want to have a connection. So we had that as a requirement. And we looked at 50 different clinical chemistries and demographics in the EMR. But I think I want to highlight one thing, right? When you're now doing in this framework where you want it to be implemented in an EMR, you want common clinical chemistries right now, because you don't want to have things that you can't calculate it in almost anyone. So we actually initially started with 60,000 people, and we reduced it to ones that had this common framework of 50,000 metrics. And so we lost about half the people, but we followed them until they died, and we estimated time till death, looking at these common clinical chemistries and other metrics of health and lifestyle that were readily available. And I'll say this here, we have a predictive model. You can do this with neural nets, you can do this with, XGBoost, you can do this in a lot of different frameworks, transformers, but suffice it to say, a predictive model where you're measuring a metric with all the data that you want. And so you're going to see what I call predictive model. Just understand you can do this in multiple frameworks. And so we did this, we modeled EMR age, we got a calculation of it using just 20 clinical metrics. And this is what I was most excited about, is it's not that many variables, right? And I'm not saying it reflects all the biology, but it's not bad to only have 20 variables that we could actually then implement over and over in the EMR. But one of the things we also have to remember is in an EMR setting, right, seldom are there points of time where you come in and you have all of the blood work done. Again, it's an antiquated system. I think we should totally change this and do micro measures and actually get a bunch of measures at consistent timeframes. But when we did this, we wanted to say, well, how robust is our measure, right? Because let's say in some people, we might not have all 20 measures at one point in time, but we may have windows of time around that we have to use clinical chemistry. And some may be perfect, others may not be. But what windows of time, to collect all the biochemistries would we need in order to create this measure? And what we actually found is whether we had those blood biochemistries in really strict windows, even stricter than what we see here, versus let's say there's one measure we don't have and it's 5 years away, we still had pretty impressively robust measures of EMR age. And not only that, we did this another way. I said, okay, let's just take 4 random time points in the electronic medical records and create 4 different completely independent models, right? If there's 4 completely independent models using the data, and I put in my data, theoretically, I should be exactly the same age from every model. But how well does it perform? And what we actually saw is this was incredibly robust. The correlations were 0.98 and up in the models, sort of suggesting that there's, you could do this, you could create independent models and they're all really getting at the same estimates. So we were really excited about that. And when we looked at measures of incident disease and prevalent disease, we saw pretty good associations like we'd expect when you look at other measures of biological age. And when we look at this in a framework of time till death, it clearly does way better than chronological age and pheno-age, which is another one where you see the healthiest people in our cohort, no one dies, and the oldest people die at a much faster rate, sort of suggesting that this is an accurate predictor of mortality. So to just sum up this part of this, right, with looking at electronic medical records today, we can actually capitalize on this and implement metrics of things like EMR very broadly and reproducibly that, if we do on this broad scale, can help us capture many things, including those like nonlinear metrics of aging. So this is what we're doing now. We're implementing it in the whole EMR and really hoping to take that paper, which may splash, and actually look at these nonlinear trajectories in larger groups. So that's a little, framework of some of how I'm thinking about electronic medical records and precision medicine. And I'm going to touch briefly on omics, and then go into multi-omics. But, with omics within the EMR setting, we also want to have data that you can connect with biobanks. And through Mass General Brigham, we have a large biobank with about 100,000 people consented with samples. We're hoping to increase this to 1 million and really do this in a much larger framework coming up in our sort of larger strategic plan. But through myself and then some work that's part of the whole biobank, I've been able to collect a lot of different multi-omic data, albeit in this case, most of this is just at one time point, so lacking sort of these trajectories, but still pretty robust data, right, on a lot of different platforms. And I'm going to be talking about some select omics. We talk about a ton of stuff here, but really talking about a few that in a few ways, maybe I'm looking at omics that may be extending beyond what has been done sort of typically. But I want to start with what's the logical place to start with if I'm talking about biological aging. The first question I have is, right, can you recode your EMR age using an omic? And we could recode this with many omics. We have, but I'm going to be talking about it with DNA methylation and recoding this with DNA methylation, because that's sort of where a lot of these aging clocks or biomarkers are being done. So again, we can do this in a very simple way where we have EMR age and we have our DNA methylation data and create our elastic net of measure of EMR age. So I'm now calling that DNA M EMR age, but just the predictor of EMR age. And as we look and we see, we can actually get very robust predictions and testing with high ICCs in doing this. So the point is, right, we can translate EMR age into an omic. And we can actually do that very well with proteomics, metabolomics, and DNA methylation. And they all do relatively well. DNA methylation is probably a little bit better, but they're all pretty impressively high with correlations like with correlation of like 0.89 is what we saw in all of them. So I want to just sort of pause and say, like, okay, this is great, but let's ask this question: what type of value can we get out of omics? And we saw this a lot, and the answer is a lot from the previous talk, but I want to talk about a few sort of select ways that we've done this, just to give a few snippets of perspective. And one is, okay, now that we have our DNA methylation measure of age, can we actually look at aging in the metabolome? And the answer is yes, we can look at aging in the metabolome with DNA methylation as EMR age as our outcome. But I want to talk about it in some other frameworks. because that's sort of a framework we all think of it, right? Okay, you look at the metabolome and you look at outcomes of biological age, and there's a lot of stuff we can capture there. There's a lot of major metabolites, ergothioneine. There's certain key ones that just jump out at you, reduced cortisol and androgens over time. But let's talk about it in a different framework, because this is a type of way that I think we can extend this. And one is thinking about what we can infer from the metabolome and dietary patterns. And I've done a lot of work with collaborative efforts from people at Harvard School of Public Health, Frank Hu, Li-Ming Liang, who have actually done this and actually created profiles. You can do like 8 different dietary profiles: Mediterranean diet, good vegetarian diet, bad vegetarian diet, overall vegetarian diet. You can go on. So we have about 8 of those we've looked at, and they are actually pretty impressively robust. And part of the reason they're robust is because when you look at something like the metabolome, there are key biomarkers that you can see for things like omega-3 fatty acids, animal fats, caffeine, alcohol, fruits and vegetables, vitamins. And those, while maybe other things may be changing, are pretty impressively consistent and can actually capture these profiles. But I'm actually going to even talk about it in a different framework here. And this is work I'm doing with Julia Manchetti, who is part of this foodome effort. She's one of the people leading this. And we not only have looked at these in terms of dietary patterns, but what we've done is we've looked at the chemical structure of an enormous amount of metabolites in the body, and that have been annotated to different foods. And we've actually looked at this from a structure of actually looking at all of the chemical structures to make sure we are annotating these correctly. Now, when I say annotated to food, though, there's ways that are important to think about that. So hold that thought. But I just have here just a really quick heat map of the fact that if you look at EMR age and DNA EMR age, or you look at what I'm going to talk about next, omic age, and or other ones on the market, you can really see pretty impressively that some things reduce biological age and other things increase it, and some really, really increase it, right? But I want to think about this. When we do this in epidemiological studies, there's things that need to be parsed out. And so here actually is a perfect example of caffeine, which is one of my favorite, coffee, one of my favorite things to think about. And these are sort of common coffee metabolites. But what is interesting is if you look at this and you were looking to annotate this just by epidemiological studies, what also gets annotated with it are things like cream and sugar, right? Because you eat them together, right? So if you do this in a framework and you look at the data of what's been published, my point is there are things annotated to foods that aren't actually anything to do with foods. They're common associations. And actually, if you look at this in a network, for example, you'll see a lot of the good omega-3 fatty acids. And one of the strongest things associated with it is rice metabolites, because they're eaten together. And so those are things we need to think of when we think about causation of things. But so one of the ways we parse this out with Julia, which I think is really, really neat, is we looked at different types of food, and she's actually created an entire library in the foodome, and we're continuing to expand this. And we look at types of foods, and we don't just say, okay, what's a biomarker of shellfish? Because the answer is, if you look at something like whether it's shellfish or whether it's cereals, one biomarker may be able to somewhat distinguish it, but there are good and bad things in most foods we eat, right? If we eat chicken thighs, right, there is good protein, there is bad animal fat. And those things often do not get parsed out. And that is really important. And so when we look at things like associations here with DNA, EMR age, what we see is each of these foods, these are the associations of metabolites annotated to that food. And if they're orange, that means they're FDR significant. Blue is not significant. And what we can see is for a lot of these things, right, there's good ones and bad ones. And it's important to recognize that, right? Now, what I have in X's in the middle are the median beta value of the coefficient, if you put all of those together in association with aging. But I will also say here, though, is with each one of those, while there's a range of metabolites that you can annotate to it, what we still have to do is weight those, right? Because if something has a lot of the good metabolite and barely any of the bad, right, we have to balance those out. But you can see here that with a lot of these foods, you still see the things that we know are bad are bad. Now, coffee is interesting here, and I wonder if some of this is going to tip on sort of what happens with coffee metabolism at points in life. But if you take those out and you reorder this only by what things are FDR significant, these slightly shift. But again, you see the worst things here are sort of what we'd expect. Sweetened beverages, processed food, juices, cereal, animal fats. Yet, even within some of these, some things may actually be healthy. So I think it's important to sort of understand this when we're eating foods, that there might be primary biomarkers, but we still often have good and bad with foods. So another area, and I'm just giving a few select examples of omics that I don't think have been really presented prominently. And another one that I'm super excited about is the area of viral antibodies and autoantibodies, because I think a lot of work needs to be done here. This is a very, an area that really, I think there's a lot more to learn. And I'm doing a lot of work now with Ben Larman and Joe DeRisi on looking at viral antibodies and autoantibodies. And when we look at this with aging, and I will say this is a new area for me, but I just want to highlight one really interesting thing in some of the initial things we've seen with viral antibodies. And what you see here is actually this Manhattan plot. I want to make sure I reference this. Each one there is a different biological agent clock. So we have DNA methylation, right? We've imputed a bunch of different biological agent clocks. And then these are the viral antibodies associated with biological age across the clocks. But one of the things we noticed here is with this retro clock, which is a clock that has been made looking at HERV genes, which are retroviruses embedded in the human genome from a long time ago. So sort of really a framework that is generated using retroviruses. We saw a huge number of associations with biological age. And what was really interesting in this association with this retro clock is that there are sort of two prominent things we see with viruses and aging. And one is there's certain viral antibodies that really accelerate aging. The one that was most prominent here was HIV. But in looking at this, viruses that stay in our body, so viruses like hepatitis and herpes, those seem to have a much more damaging effect on biological age. On the other side, vaccines, and we're actually looking into really annotating this really carefully. Some vaccines look like they're really consistently associated with reducing biological age, which is pretty interesting, right? We haven't actually seen that, and even though these are really high, we'll say that the p-values are more like 10 to the negative 8 here. But hepatitis V vaccines super protective against biological aging. And the last thing I want to talk about here, and I'll go through this quickly, is sort of a little bit with metabolomics and clinical implementation, just because I think this is a good example of implementation. And this is work that is coming out from Nightingale and what they've been doing in the UK Biobank. You know, one of the things they've done is created metabolic risk scores. We have risk scores for all of our favorite omics, but you know, they've done this for a bunch of different diseases. I have 12 different diseases here. And this is work done in the UK Biobank, but one of the things we asked is, is that actually really clinically applicable in the setting of an EMR, which is a different patient population, and what do we see? And so we looked at what they had sort of looked at, these risk scores and risk for diseases. And this over here is our biobank of 47,000 people. And one of the first things we saw was like, there's a really pretty impressive replication of these risk scores for different diseases. And so this is important because it does show, you know, this could be very relevant for clinical implementation. But when you have an EMR, I think there's a lot of other things you can do, not only just looking at one disease outcome, but you can look at it in a spectrum of disease outcomes. So one of the things we did was look at these metabolite risk scores and just look at them at related diseases. And this is even nice because we see that they sort of extend to some related diseases, particularly for liver diseases. And actually COPD is one of my most interesting examples, because when we looked at these different hazard ratios, when we look at this risk score for COPD, we see this huge range of other lung diseases, including asthma, and they all go in decreasing order, but allergic rhinitis was not associated, which to me was great, because COPD is a Th1-driven response primarily, where something like allergic rhinitis is the complete opposite. And while they may both have comorbidities in the lung, you shouldn't see it. So I was excited to see that. And similarly, we see pretty consistent associations with myocardial infarction. But we also see this comorbid with other diseases. Now, one thing I'll pause here. This is great because we see, it's generally reproducing. But I do want to highlight it's not specific. And this gets to the point that was made earlier. I think some of these platforms, and this one, it's a great platform in that it's super inexpensive. We can really implement it really quickly, likely on large scales. But for some of these, it's not specific. And I do think that's where other biomarkers being added to this could be really critical to make this much more specific, using other omics or even more metabolites. But then here's one I'll highlight. Colorectal cancer does not do well. And so I think there is definitely things that do need work. But the point is, it's nice to see that with a lot of these, we're actually at a point where we might be able to implement this very quickly in EMR settings if everyone gets them done. And it's inexpensive, right? It's an inexpensive test. So a lot of times I think, single omics, right? There's a lot of meaningful things we can extract from biology. We already are at a point where we can develop some cost-effective biomarkers, and there are beginning to be some success stories. But I now want to go more to multi-omics, right? Because I think this is the framework that all of us think of. But when we think of multi-omics and omics, I just want to pause and say, what's the data we want? And again, I just preceded the talk that showed all the data we want. And the answer is, we want everything, right? We don't just want electronic medical records, clinical diagnoses, clinical labs. We want digital data, right? Like, we need to move to the 21st century. Let's get our Oura rings and have them connected. Let's get our smartwatches and have them connected. We want lifestyle data, right? We want data on what people are doing on a day-to-day basis, their nutritional habits, their exercise habits. That is something that could be done inexpensively, almost for free, and we aren't doing it. So that is complete data that is at a loss that we could be capturing on a broad scale pretty easily if EMR systems had simple ways that people could consent to share some of the wearable health data. And I think if people knew how easy that could be with AI to do that and optimize their health, they would do it because it's not that hard, right? It's a very, from a point of view of cost, very low cost and very low threshold, and something I think we need to do. But let's talk about the molecular data, right? Because there's the molecular data, and this is where a lot of research is. And there's the DREAM, get the genome and the epigenome and the transcriptome and the proteome and the metabolome, the microbiome, and every exposome, right? And let's take the phenome and let's put them all together in this framework where we do systems biology. Sorry, that last little part didn't come out quite right, and we get personalized treatments. This is the dream, right? And we want to do this. And the thing I want us to highlight about this, though, our omic data are very complex, and they need to be simplified for a clinical setting. And the dream is all of the data. The electronic data, a lot of the stuff we can wear with wearables, that is really feasible. But the dream in thinking about this also comes with a cost for a lot of the omics. If you do all of these omics on a regular basis on everyone, that is a lot of money. And that's just not feasible in the framework right now of the EMR system. I think there's ways we could really reduce it. And I think that could be, a lot of the stuff you're talking about where we actually look over all the omics and have like very inexpensive, smaller panels that we develop. That's like a no-brainer thing that we should be doing and implementing. So I want to highlight that now because it's really one way to overcome this. But let's just talk about other ways that we can overcome this, even with what we have now. And thinking about the fact that right now, this isn't a reality of getting all of the omics on everyone, like once every 6 months. It's too expensive in the framework we have for everyone, particularly underprivileged, low socioeconomic, who are some of those in most need. So in thinking just about how are ways that we simplify this, right? And one way that I think to simplify this, that is a way that we've been working on, is what I call translating things to a common language, right? So if we have large number of omics now on a large group of people in research populations, the advantage of that is we can using different, imputation algorithms, AI, from one omic to the other. We can translate things back and forth. And fundamentally, that is really, really important because if it's done well, it can dramatically reduce costs. So in what we did here with omic age, what we did was we tried to focus on this framework of making the complex simple by using all of the omic data we have and translating it into the common language. And in this case, that's DNA methylation. And so again, a generalized framework where we take each metabolite, each protein, each clinical measure, and as we extend on viral responses, this and that, and can we actually create reliable predictive measures of those using DNA methylation only? And why is that? Because then theoretically, you can go in and you can just measure one thing and you can impute everything else. Now, I do want to highlight this needs to be done, and if you're doing this, where you're identifying very reliable predictive biomarkers, and you're not going to be able to do that with anything. I think the best framework, right, is to have a bunch of things that we've sort of narrowed down to. That's this panel that we measure to start with. But in taking this framework, as just an approach, that is one thing we did with the data that we had with proteomics, which is a SEER proteomic platform, metabolomic data from Metabolon, DNA methylation, 850K, and clinical variables. And we've now done let's say 80 different clinical labs and biochemistries. And we translated them all into surrogates using DNA methylation, right? So here now you see, in this case, what I'm showing are, we took the proteins, the metabolites, the clinical data that had an association with biological age, with EMR age, and then we created DNA methylation surrogates. Our ongoing work right now is actually really refining these. So we've now taken this and really refined these by looking at 10 different AI models to see which ones perform the best. But then what we can do is, right, we have our surrogate measures of the things that are important, surrogate measures of steroids, surrogate measures of caffeine, surrogate measures of ergothioneine, all these things, and measure that could be very clinically relevant, right, with clinical measures, HbA1c, glucose, triglycerides, important proteins, albumin, CRP, et cetera. And we translated this all into DNA methylation. And here are some of the correlations. But then the important thing then is we can then use a predictive model to predict EMR age, where we now have all of the data put into a common language. It only needs to be measured by DNA methylation, but it's this multi-omic informed measure of biological age, which we will call omic age. And how does omic age perform? Well, we see it performs as well as just DNA methylation or better. And then, but moreover, and we've now extended this to several other cohorts, what are the associations we see with prevalent and incident diseases? And, here we see really impressive performance of increased, of high odds ratios and incident and prevalent disease with the major sort of outcomes we would be interested in. And when we've extended this to other populations, we really see, I think, very consistently, these are among the top performers along with GrimAge. So I want to give GrimAge a shout out. GrimAge in some cases, particularly COPD, does better, but we see this very consistently. Whereas with COPD and ours, we're not quite as good. But what's the power of this now, right? Now with DNA methylation, what we can do is we're really creating more and more accurate imputation. We can inform ourselves on all of these other omics. We can inform on a lot of the different metabolic processes, regardless of whether the measure was actually even used in omic age. We have them all for everything, right? So we have a way to sort of impute this back with really then informing on biology. Not only that, when we look at this with, when it comes to measures of prediction, we really see impressive 5-year and 10-year mortality predictions. And these we've now seen robust in other studies like the health and retirement study, the same thing, that omic age in particular really stands out as being really impressively robust and a strong predictor of mortality. And not only that, now this is sort of data we have among 3 different cohorts of just looking at lifestyle factors along with omic age. And now the interesting thing about these 3 studies is they're very different. Like the biobank, our biobank is a sick population. the true diagnostic cohort, who is our sponsored research collaborator in this, is a uber healthy population. And the health and retirement study is like a normal, sort of a normal aging population. And so even when we look at a lot of these measures, like here we see underweight is associated with increased age in the biobank. Well, that's not surprising in our biobank because it's probably an indicator of people who are getting really sick, and it's probably an indicator of frailty. So there's things that we can see in terms of parsing out differences in the different cohorts. But by and large, I think when we reflect on multi-omics, right, multi-omics is certainly important for biological understanding. And there's a lot of ways we need to look at it. And I think that is the most important framework to creating an overall panels that are important of extracting a select set of biomarkers for health. But altogether, it's super complex. And we need to be thinking about how we can translate that, because if we translate all of the multi-omics, and it's just not a practical framework right now for our implementation in an EMR or clinical setting. And it would be burdensome on the patient too, right? If they have to get every single thing at multiple time points, and it's a lot of money. I think ideas of doing this where you can extract a lot of things at home, get stable biomarkers, is absolutely a way to go. But we also want to think about really the power of imputation and how that can do things and offer a compromise and be cost effective. So with that, this is just a beginning. It's really an exciting area to be involved in, and I'm excited to see all the things that are coming up day to day. But I think getting to this framework is feasible. There are certain things in regulatory things that also need to happen, but I think it's a framework that we're now really having a strong vision for. So I'd like to say thank you and thank the people on my team for all their work.